Good evening, good morning, good afternoon, everybody. It is actually 8 p.m. in Paris, 7 p.m. in London, 2 p.m. in Hamilton in Canada. And it is my great pleasure to welcome you to this new journal club brought to you by the International Academy for Clinical Hematology. I'm Mohamed Mouti from the Sorbonne University and Saint Antoine Hospital in Paris, in France. And I'll be joined today by a very distinguished panelist, myeloma expert, namely Dr. Hira Mayan, who is an assistant professor at the McMaster University in Hamilton. As you may have seen in the announcement of the Journal Club, we were supposed to have Dr. Marivi Mateos from the University of Salamanca as a panelist because she is the co-author of the papers we will discuss today. Unfortunately, only a few minutes ago, Dr. Mateos had to cancel her participation because of a last-minute issue. Nothing very bad, rather very good news for her, but I can't mention this right now. And actually, to be honest, we have launched this journal club now more than 2 years ago. And until now, we never had such experience of a last-minute cancellation like 30 minutes before the broadcast. And I was always wondering when it is going to happen. And indeed, well, there is a beginning for everything, but I'm very glad and very grateful that Dr. Mayan accepted to kindly participate without the other panelists. And I'll do my best to play a sort of a double role, the moderator, but also a little bit of a panelist in the field of multiple myeloma. So now I'll switch to my, I would say, moderator role, and as usual, give the introduction of the journal club. The usual acknowledgement to all our supporters who are really allowing us to deliver all of these activities free of charge across the globe. This is Hematology Without Borders. And of course, you all know that beside the Journal Club, we have the Junior Club about case reports presented by our younger colleagues. We have the monthly webinars. We have the Giants series. We do have the IACH for nurses. We do have our publication, Clinical Hematology International, now referenced on PubMed, and many other activities. So big thank you, very warm thank you to all our supporters for their unrestricted educational grants. So the Scientific Steering Committee of the IACH has decided to choose for this journal club actually two papers. Again, this is quite unique because usually we go with one paper, but these two papers were actually published very recently, a few days ago, in the same at the same time, and they were both about a novel agent in multiple myeloma, namely Melflufen. And the first paper was the so-called Anchor trial, and the title was Melflufen plus Dexamethasone and Daratumumab or bortezomib in relapsed refractory multiple myeloma, final results of phase 1/2A study. And this was led by Dr. Enrique Ocio, also from Spain, with a co-author, Dr. Mateos, who was supposed to be our second panelist today. And the second paper is the LIGHTHOUSE study. And this is about the efficacy and safety of Melflufen plus Daratumumab and dexamethasone in relapsed refractory multiple myeloma results from this randomized open label phase 3 Lighthouse study. And obviously, because of the for the sake of time, I will not go through all the details of these papers, but I will simply try to summarize the key messages, and then we'll open the discussion with Hira. But also, this journal club is brought to you live, so please don't hesitate to post your questions and send your comments. So let me start first by the summary of the first paper that we have chosen, namely the Anchor study. And this is a phase 1/2a study which evaluated melflufen plus dexamethasone with either daratumumab or followed by bortezomib in triplet combination with daratumumab or bortezomib in a patient with relapsed refractory multiple myeloma. And I have highlighted this in yellow. And as you may guess, it's a phase 1 trial. So the goal, the primary objective actually, was to define the optimal dose of melflufen in such a triplet combination, but also, of course, get a sense of the overall response rate in the phase 2 part of the study. So we had 33 patients treated in the daratumumab arm and 23 patients treated in the bortezomib arm. And the good news that there were no dose-limiting toxicities at either melflufen dose level. They used 30 or 40 milligram. And with both triplets, the most common, more than grade 3 treatment emergent adverse event were thrombocytopenia and neutropenia. And I believe we'll come back to this in the discussion because, of course, of the nature of the drug melflufen, which is a first-in-class alkylating peptide drug conjugate. And to make a long story short, the 30 milligram dosage of melflufen based on the phase 1 and early trial was established as the recommended dose for use with dexamethasone and daratumumab or bortezomib for future studies in relapsed refractory multiple myeloma. The response rate were more than 70%, 73 and 78%. And you can see the PFS figures between 13 and 15 months. But again, we are in a phase 1/2 trial, and the primary objective was about really finding the dose. The second paper is dealing with the randomized so-called LIGHTHOUSE study. And the reference of the study is here. And this study assessed melflufen plus daratumumab and dexamethasone versus daratumumab in relapsed refractory multiple myeloma patient, in those patients who were refractory to an immune modulatory agent and a proteasome inhibitor, or who had received more than 3 prior lines of therapy, including an immunomodulatory agent and a proteasome inhibitor. There was something very specific about this study because initially it was put on hold, and this led to financial constraints. So this is about the complexity of running these large studies. And unfortunately, the study was closed prematurely. Nevertheless, and the investigators needs to be congratulated for this. They were very resilient, I would say, and they provided the analysis of the patient who were included, although the study didn't go until the end. And I believe this is something very important because our duty, our role as academic investigators is really to make data available, even if things didn't go exactly the way we wanted initially or we planned them initially. And the conclusion from the study, from at least the group which was analyzed, because obviously it was difficult to draw any final conclusion when you have stopped prematurely the study, that melflufen plus daratumumab dexamethasone demonstrated superior PFS and response rates versus daratumumab alone, with a safety profile comparable to previously published melflufen studies. And of course, I would like to anticipate, I'm sure, some of the questions we will receive about how to design an optimal trial and what would be the control arm. But again, guys, things needs to be analyzed in their context when they have been designed, because obviously, retrospectively, it is always much easier to design the ideal trial. So I'll stop this, and I would like to welcome again Dr. Hira Mayan. Thank you, Hira. I know you have a busy clinic, but you managed to free some time for us. So I'm very grateful on behalf of the IACH team. So my first question to you, Hira, and I think the participant would love to hear from you, whether you can provide us first with a sort of a summary about what is melflufen, because I'm not convinced everybody is aware of this agent, which is, by the way, approved in Europe and can be used. I don't know in Canada for you, but in Europe it is approved, definitely. So can you give us a brief summary about this agent and its mechanism of action?
Thanks, Professor Moti, and thank you so much for the invitation. And again, thank you to the International Academy for Clinical Hematology for leading these journal clubs. I know a lot of us junior faculty rely on this, so thank you so much. So, melflufen, I guess the first thing to say, it's different than melphalan. It's really a kind of a first-in-class alkylating peptide drug conjugate. And it's unique in its factor that, it takes that cytotoxic payload and delivers it into the tumor cells. So that's kind of traditional for how different than the traditional kind of large cytotoxic agents that we think of. So it's really a much more targeted and different agent, and really a first-in-class molecule of this kind.
Excellent. So that brings us actually, Hira, to the issue, and I believe, and here maybe there would be a little bit of divergent opinion between both sides of the Atlantic about the value of alkylating agent. Obviously, this is like the next generation of alkylating agent, and we in Europe, we believe that alkylating agents remain a sort of a backbone of the treatment of multiple myeloma. So what is the situation for you in Canada? And I personally believe that we need these next generation alkylating agents to be developed.
Absolutely. So, I would say probably you and I are closer in thought around that compared to my US colleagues, just because of all the different agents that they have. So alkylators, again, remain part of many treatment regimens. They're a great add-on drug as well to a lot of existing regimens where, by adding an alkylator, and again, we're mostly used to adding cyclophosphamide as a old school alkylator, we can get a little bit more in terms of response rate, get a little bit more in terms of the durability of response. So I think, as we think of a lot of these novel compounds and certainly immunotherapies taking over the field and fantastic, I actually still think there's a lot of strength in thinking about these traditional and newer generation kind of cytotoxic agents like cyclophosphamide, melphalan, and even melflufen, because again, they have, different type of action, and they're different from a lot of the therapies that are being developed. And I think there's probably a lot of role, like, the LIGHTHOUSE trial tested, of combination strategies with these, because again, these are drugs that we're well used to working with, and certainly, hopefully, with these next generation of compounds like melflufen, that we can have some decreased toxicity as well with some of these agents.
Yeah, no, thank you very much. And I think that brings us to the question, which is a little bit broader than the specific case of melflufen, and why do we need these novel agents like melflufen? Because I always hear from time to time that, oh, in myeloma you have so many drugs, why do you need so many options? And I always reply that until we have cured every single patient, I'm more than happy to take any option and validate it and make it available. It doesn't mean that every single patient should receive drug X or drug Y or drug Z, but maybe here, can I kindly ask you to comment on the current situation where we see more and more patients not only becoming triple, but even penta-refractory, but even recently I've seen patients who are, I don't know if we can say exa-refractory or something, because they have even failed the CAR T cells,
I know. And I think, Professor Moti, you bring up an excellent point. When we have a 50-year-old patient in clinic, telling them that the average lifespan with myeloma, 10 years, 15 years is not good enough, right? We want this to be much longer than even where we are currently. And with all of the newer therapies, moving more up front, you could have a patient by second line, who's certainly triple class refractory. And you're right, now we're seeing, I guess, patients who are BCMA refractory, and the options for those patients, again, are so limited. And as we move these therapies up front, we will have these patients, in our clinic in the next year or 2 years who've seen an immunomodulatory drug, proteasome inhibitor, anti-CD38, and BCMA, and we're still going to need more options. So I think certainly developing, either kind of thinking of these newer generation cytotoxic agents or developing newer therapies, we're going to need it, because a 10-year survival for, again, a 50, 60-year-old patient is not adequate. We still have a long ways to go in myeloma.
Yeah, absolutely. And I think there is now more and more an agreement on this. And that brings me actually to the issue of whether is it relevant today to combine a new agent being developed with daratumumab, because one of the criticism we heard about these studies is that they are being combined with daratumumab or being tested against daratumumab single agent. And today, I believe, at least in those countries which can afford it, most patients are receiving actually daratumumab very early in the course of their disease. So that, and obviously, as long as they are responding, probably they will not switch to another line of therapy. But if they become resistant to an anti-CD38 monoclonal antibody, then may wonder whether we should continue developing drugs in combination with daratumumab. So what do you think? What's your opinion? I know it's a $1 million question, because when we start a trial today, when we start developing something today, we need to think actually about the landscape in 5 or 7 years, because this is the time that and sometimes you fail and sometimes you get it wrong. And we have many examples of trials which get it wrong.
So, I mean, I think, combination therapies are still always going to be important in myeloma. We know, again, with the way myeloma is and with the tumor tiding, that we need agents that attack kind of multiple targets at the same time. And I guess, we need to think of what drugs can we safely combine. And so, again, Daratumumab is one because of its safety profile, is somewhat of an easier drug to combine. And again, we're seeing that with some of the immunotherapy as well. It's interesting. You're right. By first or second line, most patients will be Dara exposed and Dara refractory. But I think it's hard to know because it's possible, in the future, we may have time limited or fixed duration Daratumumab, and re-exposure to an anti-CD38 may be possible after you've been off of it for a little time, or perhaps if you aren't refractory to it. So I think we'll have to think about how to use these agents really effectively in different lines of treatment. And we'll certainly need kind of combination therapy. So, when I think of the future, can we think of a future where we use Dara up front, but maybe for a fixed duration of, again, I'm making up a hypothetical scenario, fixed duration of cycle, my patient goes into MRD negativity, there's sustained MRD negativity. I'm able to give them a drug holiday, and I'm able to reuse this drug later down the line when my patient really needs it. So I think we're going to need some kind of innovative and different types of dosing strategies that we're not sometimes maybe used to thinking in myeloma, now that we have such great, fantastic agents.
Thank you very much for clarifying this. And here I would like to answer a couple of questions about whether having in the randomized trial Daratumumab as a single agent as a control arm. And here, in my opinion, well, first of all, I agree today that in 2023, we are the 19th of September, Daratumumab single agent in the US or in France or in most Western countries, I would say, rich Western countries, is not used anymore as a single agent. However, I must confess, I was recently traveling in some Asian countries. The only approval of Daratumumab in relapsed refractory was as a single agent. So it's not impossible. Also, I'm trying to answer some of the questions we're receiving. At time of the design of the trial, actually up to 48% of the patients in Europe were receiving, because of the initial approval, Daratumumab as a single agent. So things need to be put in context. Obviously, today, between 2015 and now 2023, we have 8 years. Things have changed, and they have changed in a good and positive way. So we're really very happy about this. One question here we have from the audience here for you is, because of, I would say, this is the big group of family of alkylating agent, modern, I would say, alkylating agents, these peptide conjugate. And we know that usually chemo has some good efficacy in extramedullary disease, for instance. What's your feeling? What's your experience? And this is maybe a good niche for drugs like Melflufen, for instance.
So, I mean, those are still really great unmet needs, right? Extramedullary disease remains even, hard to get good responses, even with patients we put on bispecifics and CAR T, where either it's hard to, get them to a CAR T trial, or they're still resistant to bispecifics. So, I mean, we will still, in certain subsets of cases, need these traditional cytotoxic agents. And, alkylators, again, may fulfill that niche, likely, again, in combinations with other agents. We'll have to tackle some of this, but it may certainly be, a patient, again, who's potentially BCMA refractory with extramedullary disease, where, we're limited in our options, pulling cytotoxic traditional therapy, and again, next line, drugs like Melflufen may be helpful.
Superb, superb. Thank you very much. We have here a question from Dr. Salinas Rodriguez from Peru, and he is very loyal to the IACH activities. So thank you, Virgilio, for following us. About whether, like other alkylating agent, Melflufen can produce new genetic abnormalities, and we know that there is now more and more interest about the issue of secondary malignancies. I believe we do not have yet sufficient follow-up with melflufen to conclude whether it can produce more or less compared to other alkylating agents. However, and again, that brings me to the alkylating agent issue. The median age for multiple myeloma in the rich countries is between 70 and 72, actually. And in a standard risk cytogenetics, given the frontline treatment we give with quadruplet anti-CD38 plus VRD or KRD, followed by high-dose melphalan, followed by lenalidomide maintenance, actually, we are allowing them maybe 7, 8, or 9 years of PFS. So assuming you have a patient who is 65 or 66, you add another 10 years, or this is a patient who is now like 74, 75. You give them a salvage therapy. We had recently a publication about the long-term follow-up of the IKEMA trial, isatuximab plus carfilzomib dexamethasone. And you give them another median PFS of 42 months. Then you add a third line, whatever it is. It brings the patient to more than age 80, which is like almost a life expectancy in many countries. So why, in your opinion, suddenly we're talking about this risk of secondary malignancies and maybe overemphasizing it? I'm not saying we should not do research about it, we should neglect it, but what's your opinion? And I know there's maybe no right or wrong answer here.
Yeah, so I mean, it's a great question about melflufen and the risk of, again, long-term secondary malignancies. Again, with the trials that we so far have, we just don't have enough long-term data. And again, a lot of the, when we think of some of the alkylating agents, most of those patients have been transplanted, they've been on lenalidomide maintenance. There's a lot of confounding effects when we think of these secondary malignancies. So it's really hard to take apart one particular agent. So, I mean, certainly in my patients that are the median age, like you said, in Ontario, Canada, it's 72. It's not at my forefront. I use the best available therapy that I have at that moment. I would say, Professor Moti, when I have a much younger patient, or a patient, again, in their mid-40s, they are considerations that I do think about it, but we still know that the leading cause of death in myeloma patients is myeloma. So if I have a best therapy that controls the myeloma with an acceptable toxicity, that is what I'm going to be using. So it will be important to follow this up long term. And again, we based it with the OCEAN trial results and the impact of previous auto transplant and its outcome with melflufen. We will need to watch some of those signals long term, but I think at this point we just don't have enough data around it.
Excellent, thank you very much, and I think it's very clear. So now, from a practical point of view, assuming we have melflufen available everywhere, my best guess, and I may be wrong, is that in those patients who are in advanced relapse, physicians, but also patients would love to have CAR T cells because it's a single shot treatment and they have they can enjoy a treatment-free interval. We know that the results of bispecific antibodies are quite amazing, and we're very fortunate actually that in August, which is like not long time ago, we had 3 approvals, an extension for the approval of teclistamab, the approval of elranatamab, and both teclistamab and elranatamab are directed against BCMA, and we had also the approval of talquetamab. So bispecific antibodies are really making a big difference. But obviously, once the patient have failed these immune therapies, it's about redirecting the T cells. This is where I believe we need these agents like the XPO inhibitors, selinexor, like melflufen, like mezigdomide, published last week in the New England Journal of Medicine, the new cell mode. What's your opinion, Hira?
I think so. I mean, I think you're entirely right about this. You know, as the immunotherapies move up front, by the time our patients, maybe in a few years, are going to be second or third line, they will be exposed to really triple class refractory plus BCMA refractory, and then we'll be left with, you know, still young patients who don't have additional lines of therapy, and we will need some of these agents like melflufen potentially, or mezigdomide. I also think, you know, when we think of those patients, probably a lot of those patients will be older as well by that time. You know, when we think of a newly diagnosed patients that, again, followed the sequencing that you're talking about, we may be talking about a patient, you know, who's well into their 80s, and then we really need to think of options that are going to be tolerable, decrease treatment burden, improve quality of life, and doesn't have a lot of toxicity. And so we'll need to, you know, have some really good options that we can tailor individually for our patients, because probably by that time, you know, a number of patients will be more frail and older. So having options that allow us to choose between a huge variety will be fantastic.
Excellent. We'll take one question about the toxicity of melflufen, and actually what has been quite positive, I would say, for me from the experience with melflufen was that it's mainly hematologic toxicity. And I would say, well, obviously, toxicity is never a good news.
Yeah.
But at least hematologic toxicity, this is something we hematologists know how to handle. It's a bit more difficult when it comes to something totally different. What do you think?
I would entirely agree with you. You know, I can deal with platelets of 10, no problem, but even grade 1 eye toxicity, I have to think about. It's just not what I'm used to dealing with. And I actually think now with some of these immunotherapies where, you know, we're seeing patients with CRS and ICANS, and really our level of expertise and knowledge has expanded rapidly in the past few years. Hematological toxicity, again, is important for patients. It can impact, again, their quality of life, is associated with risk of infections, which has real consequences. But for the most part, I agree with you. As a clinician, I feel much more comfortable managing, you know, neutropenia and thrombocytopenia, as that's really our bread and butter in hematology, for sure.
Excellent. Another one question we had from Dr. Rashidi about cardiac toxicities, and maybe you can let's broaden a little bit the question, Dr. Mayan, about what are the regimens or the drugs or the situations where you would take into account the serious cardiac comorbidities and try to tweak a little bit the management of the myeloma patient.
Yeah, no, I think that's reasonable. You know, first, you know, for patients, again, presenting with cardiac toxicity, look for, again, underlying reasons, especially concurrent amyloid, to make sure, because that can really impact their prognosis. So I would say, you know, there's probably two main points where I think about cardiac toxicity. One of them is obviously for transplant eligibility. So again, no really strict ejection fraction cutoff, but certainly, you know, above a certain point or below a certain point, you want to think about it. So that's kind of the one first point where I try to see, can I really optimize it? And again, you know, we, again, still transplant the majority of patients that are going to be transplant eligible. It remains an important part of our sequencing. So even when we have patients who potentially have cardiac toxicity, we really try to see if we can optimize them to get them to transplant. And then I guess, you know, the traditional drug that we're really used to thinking in terms of, again, cardiac toxicity, not so much with the melflufen, but again, is a carfilzomib. And I think over the years, with lots of experience, we have come up with lots of little tricks. How can we pre-screen the patients, ensure cardiology is involved up front? How can we reduce some of the fluid associated with the drug? How can we monitor the patients and, potentially attenuate our doses to decrease it? But all of these drugs, again, are really important. And as much as possible, we want all of our patients to be exposed to these agents. So I try as much to optimize cardiac toxicity rather than make it an entire contraindication for a drug.
And there is also a specific question whether when it comes to carfilzomib, actually, because we know there is a clear, I would say, management plan for the cardiac toxicity of carfilzomib. What is your practical approach to the management of carfilzomib? In our hands, for instance, it's not about the dosage, but mainly about the repetition, and actually giving like once weekly high dose of carfilzomib appears to be safer, but I would love to hear from you.
Yeah, so I would actually say, our standard of care is actually once weekly for everybody. So we kind of use more of the arrow dosing and once weekly for everybody. And even then, if someone is experiencing toxicity, we try to space out the dosing. So instead of Q weekly, we might skip a dose in between. And again, with serial echoes. So, certainly we're using kind of once weekly dosing almost for everybody right up from the front, and that's just based upon to decrease some of the treatment burden.
Excellent. We have reached almost the end of this journal club. Hira, it was really a true pleasure and a very nice experience, actually, to have a journal club with only one panelist, because unfortunately, Dr. Mateos, as I mentioned in the introduction, had to cancel for a last minute issue, a good issue. But I think it was really a true pleasure. I learned a lot from you, and I believe it was extremely timely to have actually this journal club about these two articles related to Melflufen, because actually, for instance, on the 14th of September, the CHMP, which is, of course, part of the European Medicine Agency, has issued a positive opinion on a type 2 variation to extend the therapeutic indication of Melflufen based on the OCEAN trial result. But that's another trial that we did not discuss this time. It was discussed a long time ago in a previous journal club. So thank you all for joining us tonight. Thank you, Hira, for dedicating the time for this journal club. It was a pleasure. And as always, please do not hesitate to submit your ideas about topics, speakers. And I would also remind all of you guys that in our email shots since a few months now, we had the Friends Corner. So if you have any news you would like to share with the hematology community, I don't know, congratulating a colleague for a promotion, for an award, if you would like to announce one of your local meetings, if you'd like to announce whatever information of interest to the community, the hematological community, please don't hesitate to send it to info@clinical-hematology.org. But for the time being, wherever you are, I wish you all the best. Please stay safe and keep well. Thank you very much.